Hello, everyone. Thank you for coming. My name is Dr. Lucas Runyan. I'm an American Heart Association postdoctoral fellow in the Good Lab at Brandeis University, and I'm excited today to tell you about my journey as a scientist, beginning with an early interest in chemistry, moving on to an interest in biochemistry during my graduate work, and ending now where I am during my postdoctoral work, where I combine principles of biochemistry with cell biology to ask detailed questions about how the cell works. I'm originally from Michigan. Specifically, I'm from a town called Clarkston, Michigan, which is about 30 minutes north of Detroit. I attended the local high school where I fell in love with chemistry. Chemistry was always my favorite subject, and when it came time to graduate and go somewhere for college, I decided I wanted to be a chemist, although I didn't really understand what that meant at the time. So to become a chemist, I decided to go to Marquette, Michigan, and attend Northern Michigan University. Now, Northern Michigan is a beautiful university and it's located in a beautiful town called Marquette. And Marquette is wonderful. It's surrounded on all sides by beautiful nature, and it's in the middle of a great harbor, lots of swimming and boating and fishing. And one of our favorite pastimes during the summer was to go cliff jumping off of the many cliffs all around the area of the Great Lake. So, one of the most important things about being a science major as an undergraduate, in addition to getting good grades, is you have to find a research mentor. And so I asked around the campus, I cold emailed professors, I talked to my friends to see if they knew anyone who's looking for someone work in their lab. And I ended up finding Dr. Lee Recker. Uh, he's an inorganic chemist, and he agreed to take me on in his lab as an undergraduate mentee. And in Dr. Recker's lab, we would synthesize precursor molecules into new kinds of molecules, such as the ones that we see here, and then react them with metals, such as this cobalt ion we see here in the middle, to make new kinds of chemical compounds, and then study their properties. And my everyday routine is I would come into the lab and I would get the precursor molecules together and I would mix them together in, a flask, and then I would react them overnight, and then I would come in the next day, and then I would make a sample, and I would use this machine, a nuclear magnetic resonance spectrometer, which uses huge magnets to tell you the structure of the molecule you made. And then, assuming you made the right molecule, as told you by this machine, I would then react it with the metals, and then I would get the compounds. And one of the most striking things about working with metals is they make these really beautiful crystals, like we see here, these beautiful purple crystals. And this comes from the properties of the metal itself that makes this beautiful color. I had a lot of fun working with Dr. Recker, and I learned a lot, but I knew that I wanted to study the molecules of life. I was more interested in life processes. And so I decided to go to graduate school as a biochemist. And I applied all around the Midwest, and I ended up getting accepted at a place in Ohio, located in Columbus, Ohio. You might know it, it's the Ohio State University. And if you know anything about the Ohio State University, you know that they love football. Every Saturday, the uh, the town would be just completely taken over by people from all around. Thousands of people would come to watch the football games, and I would have to push through big crowds of people on my way to the lab on those days, and it was always a huge spectacle. But I wasn't there for football. I was there to do research, and so I had to find a research mentor, just like I had to as an undergraduate. In graduate school, it's a little bit more formalized, and you basically have lots of meetings with different professors, and eventually you find a couple of professors that are willing to let you come and try out their lab in the process we call rotations. So you basically spend like a month or two in everybody's lab working with them, seeing if you like the work, you like the coworkers, that kind of thing. So after doing that, I decided that I wanted to work with Professor Dmitry Kudryashov. Professor Kudryashov is interested in how cells build their internal structures. So if we think about a normal picture of a cell you might be familiar with from like a textbook, right, from your biology class, we see this big round thing with lots of organelles kind of floating in this goo, this cytoplasm. But that's not the way we thought about cells in Dimitri's lab. In Dimitri's lab, we thought about them like this. So in this case, what we're seeing is here's our nucleus again, just like we see there. But now instead of looking at all these organelles, we're looking at all these fibrous structures. So these fibrous structures are made out of a protein that forms filaments. And this protein is called actin. And actin forms the filaments, but it needs help of other proteins to bring the filaments together into these structures that we see here. These branch things at the front of the cell, these long, skinny, straight things around the sides of the cell. And so we're interested in how do cells do this? And why do cells need to do this? Well, when everything is going properly and the cytoskeleton is being taken apart and put together correctly, it allows the cell to do things like what we see in this movie. So here we're looking at a human immune cell, which is chasing a bacteria, the little tiny thing. And what it's going to do eventually is it's going to be able to catch it and eat it. And the only way it can do that is to be able to rearrange its internal structures rapidly in response to cues it gets from the environment. So in Dimitri's lab, that's what we studied, is how do these cytoskeletal structures, these actin structures, rearrange. So my project was to follow up on a previous investigation that another student found, which was that one of the proteins that help reorganize the internal structures of the cell was sensitive to small changes in pH. So what I did is I learned how this protein senses the changes in pH. And after I did that, I was able to make mutants of the protein that can no longer sense the changes in pH. And what this has the effect of is it stops the cell from being able to move properly and respond to environmental cues. And by using mutants that remove the ability of the cell to do that, what that tells us is that normally this protein needs its pH sensing ability to be able to respond to cues in the environment. And so that's what my graduate work focused on. And all the work I did was all in the test tube. But I was interested once again in going to another level, the higher biological level again. And this time I wanted to work with cells. So in order to do that, when I went for my postdoctoral work, I opted to come here to Waltham, Massachusetts, which is a beautiful city. I love the restaurants here. Me and my wife go out almost every weekend to the Moody Street and go to the different restaurants. We also love the Charles River, which is a picturesque place, really nice. Our apartment's very close to it. We go to it all the time. But more importantly, we came here to work at Brandeis University, specifically in the lab of Bruce Good. So Bruce is focused on how cells work, just like Dimitri was, but he thinks about it a little bit differently. So if we think about the Dimitri way of thinking about a cell, which is you have all these different structures that have to rearrange to allow the cell to move, Bruce instead thinks about more than just the actin structures. In this cell here, we see several types of structures. The actin structures are in blue, but we see these green ones and these red ones, which represent different networks of fibrous proteins in the cell. And when we think about all these different fibrous networks at once, we call that the cytoskeleton. But there's many different classes of networks within the cytoskeleton, and the actin network is just one of them. One of the things that we think about in Bruce's lab is, in addition to these things rearranging to be able to build structures in the cell, that the cell needs to be able to move and chase bacteria and so on, we also think about how they act kind of like a road. So that's another function of these things, is they behave sort of like the road network that connects cities together. And what's the most important thing about a road is, well, it's that they have traffic on them, right? So what is it mean for a road in a cell to have traffic? Well, the cells we study in Bruce's lab are budding yeast, which we see here on the left. And what we're seeing are these black strings within the cell. These are called actin cables. And the way actin cables grow is they start from the tip or from the neck and they move down. And then the cell will load little things onto them, little chunks of cargo, like we see in this animation here. And the little chunk of cargo will land on the road and it'll get shot up to where it's supposed to go. We call these little chunks of cargo vesicles, and these vesicles have to be shuttled up to the tip of the cell where they're delivered to this area, and then they merge with this part of the cell, and they allow the cell to grow. And this is one of the key processes we're interested in Bruce's lab. And just to show you what it looks like in a real cell, I'll show this movie now. And what we're seeing here is these little dark spots. We can't see the actin here, but we do see these little dark spots. We see them wiggle around until they find a road, and then they shoot up into the bud of the cell. And this process is essential for cells to grow and thrive. So in addition to the actin cytoskeleton, which I worked on as a PhD student, now in Bruce's lab, we're bringing in another cytoskeletal network. So think about that image I showed you a few slides ago where we had the 3 different colors of the actin with different colored networks. So in these cells that we work on, these budding yeast, they have similar amount of networks to human cells. And I'm specifically interested in studying how the actin network interacts with the septin network. So the actin network is here in red, like we saw in the last slide. But now I'm adding a new one. This is called the septin network. So the septin network is only here at the neck, as far as we know. And somehow it interacts with the actin network to help build it. So we're interested in this process. And we want to know, what is it about the septin network that helps build the actin network? We already have some clues. We're pretty sure that what the septins are doing is they're bringing in a bunch of other proteins to them, which then help guide and build the actin network off of the septin network. And so I'm going to use biochemistry, similar to what I did in my PhD, with my new cell biology system that I have in Bruce's lab, to answer fundamental questions about how the septins in these yeast create actin networks that the cell needs to live. One of the most compelling pieces of evidence that we're on the right track with this is that if you make a mutation to the septins themselves, it manifests as a problem with the actin. So if we look here on the left, these are normal cells, healthy normal cells, and we're looking at their actin networks here in black. But when I make a mutation to the septins, which are located here at the bud neck, all of a sudden our actin structures are all messed up. So what this tells us is that the septins are needed to make the actin structures. And when the actin structures aren't made properly, we see that the cells are smaller, they're sick, they're having issues growing and dividing. So the goal of this whole process is to learn about septins and actin and how they interact in these simple budding yeast. And I will then apply lessons learned to the budding yeast to the much more complicated situation in human cells. And this will shed light for us on issues that happen with these septins in human cells, including Alzheimer's, cancer, and heart disease. So that wraps up my talk. Thank you so much for coming. I hope you enjoyed it, and I hope that you learned a little bit about what it means to be a scientist. Thank you.